Good afternoon everyone. Hope you are all fine at home. Today's class is on revision of reproductive system. We won't be able to finish all the 6 lectures in 1 hour, but I will just guide you through the various question and answers concerning female reproductive system. So the first one will be on oogenesis. So you know this meiosis occurs only in germ cells. So in case of male it is going to be spermatogenesis arising from spermatogonia, or in case of female it's going to be the oogonia, which is giving rise to this oogenesis. Now this gametogenesis reduces the number of chromosome by half. So this reduction in genetic material from the diploid to haploid involves 2 divisions, you know, meiosis 1 and meiosis 2. So when you have a short note on oogenesis, you write a brief account on what is oogenesis by definition, and then draw this picture which depicts a primordial germ cell forming a viable ovum at the end. So to start with, you had a primordial germ cell which, if you remember, has migrated from the yolk sac into the developing genital ridge. There it undergoes mitosis till around 20 weeks, forming millions of primary oocyte or oogonia. They enter into meiosis, but then they get arrested in the first meiotic division, prophase 1 of meiosis 1, right? And then during adolescence or during puberty, due to the influences of various hormones, from the hypothalamus you have the GnRH, from the anterior pituitary you have FSH and LH, which helps in initiating menarche or the first menstrual period or the menstrual cycle in a female, which leads to expulsion of a single ovum every month during ovulation. So when this cycle starts, just before ovulation, the first meiotic division is completed and it enters into a secondary meiosis, right, forming a secondary oocyte with a polar body. Now this is arrested in the metaphase of meiosis 2. After ovulation, if fertilization occurs, then the meiosis 2 is completed. So you need to draw this picture along with writing a paragraph mentioning the changes which happen from a primordial germ cell to the formation of a mature oocyte, right? So this will be the difference between the spermatogenesis and oogenesis. Here you can see a spermatogonia giving rise to 4 viable sperms, but in case of oogenesis you have a primary oocyte forming a single viable secondary oocyte along with 3 polar bodies. So you need to mention the various stages at which mitosis happens, and the stage at which the first meiosis is arrested, and the stage at which the second meiosis is arrested, and the stage at which it is completed, right? So, and then end the small paragraph with writing these changes happen during the process known as folliculogenesis, which happens in the ovary during each menstrual cycle, right? So if oogenesis is a question, you define oogenesis and then go on to explain that picture starting from the primordial germ cell to the formation of a secondary oocyte, and then write a small paragraph on the various changes happening in between, right? And then end by saying that these changes happen during the process of folliculogenesis, and you can draw this diagram which is there on the right side if you have time. Now going to the LAQ of menstrual cycle, first write the definition, which is nothing but a during normal reproductive years of a female, you have a monthly rhythmical change in the rate of secretion of female hormones FSH, LH, estrogen and progesterone. Remember, you can't mention GnRH because there is a cyclical secretion but not in a monthly pattern. It is a pulsatile secretion of GnRH which is responsible. It does not follow the pattern of cyclical variation during a month, and have corresponding physical changes in the ovary, uterus and other organ. The cycle duration averages around 28 days, where it can vary from 25 to 45 days, right? So this rhythmical pattern is called female monthly sexual cycle or your menstrual cycle. Now you need to mention 3 things under this: the changes which happen in the ovary, the changes which happen in the endometrium and the corresponding hormonal regulation, right? So if an LAQ is asked on menstrual cycle, you need to mention the definition with all the 3 changes in detail. But it can also be a part of BAQ or SAQ where only the changes in the ovary or the changes in the endometrium or the hormonal regulation can be asked, right? So then first you start with the ovarian cycle. So by definition, the function of the ovarian cycle is to expel a single ovum, right, every month following each menstrual cycle from a single dominant follicle. You then divide the answers according to the following heading. You have something known as folliculogenesis, which is followed by ovulation mid-cycle. Then you have formation of corpus luteum. It can be either of menstruation or of pregnancy. And if it is of corpus luteum of menstruation, finally you have death or atresia of the corpus luteum, right? Or for convenience sake, you can divide it into two halves. You write it under two major subheadings. Activation of hypothalamic pituitary ovarian axis leads to what is known as the follicular phase and what is known as the luteal phase. So follicular phase, which begins soon after corpus luteum degenerates, that is, remember, from the previous cycle, and it lasts 12 to 14 days, and the end result is ovulation. Then after ovulation you enter what is known as the luteal phase, which begins at ovulation, lasts 12 to 14 days, and it ends when the corpus luteum degenerates. So either you divide the answer according to the previous four subheadings, or you can mention it under these two topics, right? Now you go to the follicular stage and you put a diagram which depicts a picture showing a primary follicle getting converted to a secondary follicle, pre-antral follicle, then you have the formation of antrum, so you have the antral follicle, and finally you have the pre-ovulatory follicle, which is also known as the Graafian follicle or the tertiary follicle, which is followed by expulsion of the ovum, right? You need to draw this diagram and mentioning two lines regarding each follicle, right? What is primary follicle? That is, you have the maturing ovum inside surrounded by a single layer of granulosa cell, and then it moves on to the secondary follicle where there is a differentiation between the theca cell and the granulosa cell, and then the secretion of hormones increases, so there is a formation of antrum, and how finally you have the formation of cumulus oophrys, right? All this is clearly explained in the previous lecture on ovarian cycle, right? So you draw this diagram and mention one or two points under each follicle, types of follicle, right? And then you need to write about what is theca cell and what is the granulosa cell, how the theca cell has got only LH receptors, while the granulosa cell initially it has got FSH receptors, and after ovulation it acquires the LH receptor also, right? So when we have reached till ovulation, then you need to say what are the changes, hormonal changes which brought about this ovulation. So you need to mention that starting at the beginning of the cycle, due to the previous cycle's degeneration of corpus luteum, there is no estrogen and no progesterone, which leads to a negative feedback mechanism and causes an increase in LH and FSH from the anterior pituitary. So you can show draw a graph like this which depicts the rise of these hormones, and then as the FSH is increasing, you can see that the developing follicle is also secreting more estrogen, right? There will be a selection of a prominent dominant follicle. So then you can say how the dominant follicle has started secreting more estrogen, right? And this estrogen, just 36 hours before ovulation, this estrogen causes a positive feedback mechanism wherein there is a surge of LH or the luteinizing hormone, right? So you need to write, after explaining the changes, physical changes in the follicle till ovulation, in a small paragraph mention the hormonal changes which led to this ovulation, right? So you will begin with the low level of estrogen and progesterone from the previous cycle, leading on to an increase in FSH and LH due to negative feedback mechanism. This FSH causing an increase in the size of the dominant follicle and causing an increase in the formation of estrogen, right? And just 24 to 36 hours before ovulation, this increase in estrogen causes a positive feedback mechanism resulting in what is known as LH surge, resulting in ovulation, right? After this paragraph on hormonal regulation, you go to mention a paragraph on what are the changes which happens during ovulation. So this ovulation occurs during the midpoint of every normal menstrual cycle. It is triggered by LH surge, and this LH surge is because of the onset of peak estradiol secretion, right? Now you need to know what does this LH surge cause. Three important function. One, the prophase of meiosis 1, where the primary oocyte was arrested in this stage. So that first meiotic division is completed. So it forms a secondary oocyte which is going to be expelled during ovulation. 2, formation of corpus luteum or luteinization. And 3, this LH surge causes a rapid secretion of progesterone, right? So this LH surge, 3 important functions you need to mention, right? And then you can draw this diagram from Guyton. Remember, ovulation, 3 P's are involved. One is proteolytic enzyme, other one is progesterone, and the third one is prostaglandin secretion. So all these 3 together causes enlargement of the follicle with formation of one point known as stigma, which ruptures due to this proteolytic enzyme and then releases the ovum, right? So these are the changes which happens during ovulation. Then what happens? Then the subheading comes formation of corpus luteum. So the remaining follicular, granular and thecal cells, they join together to form what is known as corpus luteum under the influence of LH. Now what is the change? Is there is a temporary shift in the secretion of progesterone when compared to that of estrogen, right? If you remember the 2-cell theory, you have this LH, this is the theca cell, basal lamina, and then you have the granulosa cell, right? So in the thecal cell we have the LH receptor, and then we have the cholesterol which forms the androstenedione. Androstenedione diffuses through the basal lamina and reaches the granulosa cell. Here the presence of FSH receptor and the FSH acting on it causes an increased formation of aromatase enzyme, leads to the conversion of androstenedione into estradiol, which is then released into the circulation. Now post ovulation there is an occurrence of LH receptor in the granulosa cell also. So you have increased formation of progesterone which is released into circulation, right? Now then you can mention 2 lines about corpus luteum. How it is programmed to live for 14 days. It reaches around 1.5 cm at the end, and then if it is no fertilization has occurred, then we name it as corpus luteum of menstruation. But if fertilization has occurred, then we use the term corpus luteum of pregnancy. This is supported by HCG from the trophoblast if fertilization has occurred. So till the placenta can take over the function of formation of estrogen and progesterone, the corpus luteum provides that. If fertilization has not occurred, the corpus luteum becomes what is known as corpus albicans, that sinks into the middle of the ovary and it is slowly observed. And now because it is now degenerated into what is known as corpus albicans, it no longer forms estrogen and progesterone, right? So this forms the end of the cycle, leading on to a negative feedback mechanism causing an increase in FSH and LH from the anterior pituitary, right? So this is a summary of the ovarian changes. If you have time, then you can draw this showing the various stages finally leading on to what is known as a degenerating corpus luteum or a corpus albicans, right, which gets incorporated into the interstitial tissue. So this is about the ovarian changes. First is define the ovarian what is the end result and what are the 4 stages, and then draw the diagram of the different stages of the follicle. Then the changes happening during ovulation, right, and then formation of corpus luteum, and then whether it degenerates to corpus albicans or it forms corpus luteum of pregnancy, and write a small paragraph on the hormonal changes which lead on to the formation of this, right. So these were the ovarian changes and the hormonal changes which happen, and it is better to draw this diagram which depicts the hormonal changes. So beginning of the cycle you have an increase in the FSH and LH. This FSH increase causing an increase in estrogen from the dominant follicle. So before ovulation you have an LH surge, and that is due to this estrogen surge, and post ovulation you can see the progesterone which is more compared to that of the estrogen, and slowly due to these 2 things are more, negative feedback mechanism, your FSH and LH are less, and that depicts the end of the cycle. And here you can see the corpus luteum degenerating into corpus albicans. So there is no longer estrogen and no longer progesterone. So by negative feedback mechanism, the next cycle starts with increased formation of FSH and LH, right? So now then you start writing about the endometrial changes, or known it as the uterine cycle. So here you divide it into three important phases. You have menstruation, which means sloughing or shredding of the stratum functionale layer of endometrium. Then you have what is known as the proliferative phase, which is again known as the estrogen phase of the endometrial cycle occurring before ovulation. Then you have the secretory phase, which is the progesterone phase, which happens after ovulation. Brief anatomy. Remember the part in the lower end is known as stratum basale, and the upper portion is the stratum functionale, which is shed during each menstrual cycle. And remember the spiral arteries, and then with this part is the stratum functionale, which is shed during menstruation. So the beginning of a new cycle, you have what is left over is the stratum basale, right? So see, ideally the menstrual cycle or the endometrial changes, when I say it's a 28-day cycle, the first few days are menstruation or sloughing of the stratum functionale, right? By 5th or 6th day, the proliferative phase starts, and after ovulation, the secretory phase starts. So it is menstruation followed by proliferative phase followed by secretory phase. But for easy understanding and to make it more presentable, what we do is we start the answer by writing the proliferative phase, estrogen phase, or the pre-ovulatory phase. So now here we have assumed that the first part, 5 to 6 days, menstruation has happened and there has been sloughing of the tissue of stratum functionale. So what is remaining is your stratum basale, and we know that by now due to the action of FSH on the granulosa cell, there is an increase in the formation of estrogen, right? Now this rising level of estrogen during the follicular phase of ovary induces the proliferative phase of uterine endometrium, right? So what are the changes which happen in the endometrium? Everything is getting proliferated. You have proliferation of basal stromal cells, epithelial cells, you have the glandular component as well as the stromal component, which is showing increased mitotic activity. Plus there is cellular hyperplasia with increased extracellular matrix, right? All this happens because of estrogen acting on its ER receptors, bringing about a gene expression along with growth inducing factors, right? So this changes in the endometrium due to estrogen. First 4 to 7 days there is rapid proliferation, then 7 to 10 days there is a growth of stromal as well as epithelial tissue and new vessels being formed. So finally at the mid-cycle, just before ovulation, from 0.5 millimeters, which was the thickness at the beginning of the cycle, it has become 5 millimeters thick, right? So this picture shows this is the early proliferative phase, right? And this is the late proliferative phase just before ovulation. Again the same histological picture. This is immediately after menstruation and this is the proliferative phase just before ovulation, right? Now what happens, ovulation has occurred, so there is a shift in the level of importance of hormones, because now you have the corpus luteum producing high levels of progesterone along with estradiol. Now this progesterone does not cause much growth, but it induces the differentiation of the cells, epithelial and the stromal cells, right? So you have the progesterone which is stimulating the glandular component, so the endometrial glands become engorged and tortuous, and remember there is increase in the vascularization of the endometrium. Now this shift happens because assuming fertilization has taken place and the blastocyte has to get implanted in the uterus, and the uterus has to provide the nourishment for the growing fetus. So these changes which happen in the endometrium, increase in glandular tissue, increase in secretory rich fluid, plus increase in vasculature, we use the term what is known as pre-decidulization, right? So under the influence of progesterone, stromal cells change into what is known as decidual cells. Now this uterine glands, there is an increase in the nutrient rich product, especially glycogen, that support the blastocyte viability. Now the glands become corkscrew and sacculated. Now this layer is known as zona spongiosa. Now you have zona compacta and spongiosa forming what is known as functional layer. So which is going to be shed during the menstruation if pregnancy has not occurred. You can see this, the glands changing into corkscrew shape, right? Here again with rich deposition of glycogen in the stroma. The thickness is the same, almost the same, but there is a differentiation in the type of tissue there, forming what is known as decellulization. Right? Now, how does this progesterone decrease the biological activity of estradiol? It reduces the number of receptors. It increases the enzymatic activity of 17-beta HSD. So, where estradiol is getting converted into estrone, and there is increased activity of estrone sulfuryltransferase also. So now we go back to menstruation, right? Ideally in a cycle, menstruation depicts the first 4 to 5 days, but for understanding purpose we have dealt with proliferative phase, we have dealt with secretory phase, and now we have come to the menstruation phase, right? This starts when you know that the corpus luteum has got degenerated. The ideal life cycle, we say it's somewhere around 12 plus or minus 2 days, that is 14 days. Now once the corpus luteum has degenerated, there is a decrease in the formation of estrogen and progesterone, right? So in this scenario, what happens is the spiral arteries rhythmically go into spasm and then relax, forming what is known as the ischemic phase. Now when there is no blood supply or the blood supply is compromised to the stratum functionale, the cells begin to die, and you have hydrolyzed enzymes which are released from the lysosomes and which causes the breakdown of endometrium, right? So prostaglandin production is increased. At the same time you have necrosis of vascular cells which leads to microhemorrhage, right? So all this together leads to the loss of zona functionalis or stratum functionalis. Now remember, this coincides with the early follicular phase of the ovary. So as the hormonal support of the endometrium is withdrawn, the vascular glandular integrity of the endometrium degenerates. So finally you have the tissue breaking down and the menstrual bleeding ensues, right? So this is the day 1 of the menstrual cycle. So then you need to write a small paragraph on how uterine contraction causes the expulsion of uterine contents. So the average loss is somewhere between 30 to 70 ml. It has loss of blood, tissues as well as the serous fluid. The duration normally is somewhere between 3 to 5 days. Right? Remember, the menstrual blood does not clot due to the presence of fibrinolysin released from the necrotic endometrial tissues. So finally you end the answer. If you have time, you can draw this picture depicting all the 3 changes. Below down is the hormonal change. You divide the page into either half left and right. The middle portion depicting the period of ovulation or the 14th day, right? And drawing, remember, you start 1 to 5 days with menstruation, then you go to proliferative, then you reach the secretory phase. Though while writing we started with the proliferative phase, secretory phase, and then we ended the answer with menstruation. That we followed that sequel for your better understanding. But otherwise, when you're depicting it in a picture, remember the first few days it is the menstruation, then which is followed by the proliferative phase, and then which is followed by the secretory phase, right? So you write the changes, folliculogenesis, the luteal phase, right? In between you have the ovulation period. Then you draw this diagram which shows the changes in the endometrium, and finally the hormonal regulation. So this is about the LAQ on menstrual cycle, or it can be part of short notes where they ask you only the ovarian changes or only the uterine changes. Remember, it is important to know the changes in the cervix also. So again we divided into 2 phases. The first phase where the estrogen hormone is more, or the proliferative phase. Here remember, the secretion is copious, clear, elastic, thin and alkaline cervical mucosa. This is ideal for the survival and transport of the sperm, right? Now this stretchability or elasticity of the cervical mucosa, it is evaluated by taking them in between 2 glass slides, and we use the term the spin bucket egg white quality, right? Because of the increase in the elastic content. Now this also has increase in sodium chloride. So when it is dried, the cervical mucus is dried on a slide, you get what is known as fern pattern. These two are diagnostic of the estrogen phase of the cycle or cervical secretion. Now after ovulation, during the secretory phase or during the progesterone phase, what happens is the mucus is scanty, thick, acidic, tenacious and cellular. It is understandable because this becomes hostile to sperm, assuming that fertilization has occurred, so it does not want the other sperm to enter. So the mucus becomes scanty, thick, acidic, tenacious and more cellular, right? So this is sometimes they can add a note on the cervical changes during the menstrual cycle, right? So you start with definition and you say that there are three important things which are happening: ovarian changes, hormonal regulation, and there you have the endometrial changes. And then start writing about the ovarian changes, divide that into follicular phase or luteal phase, and then explain till the formation of corpus luteum, and write a small paragraph depicting the changes in the hormone which brought about these changes. Then go on to explain the endometrial changes, draw the diagram which shows menstruation, proliferative phase and then the secretory phase, and then start writing a small paragraph under each subheading. Then you can finish the answer by drawing a diagram which shows all the three. Right? Another important short notes is the fetal placental unit. Remember, there are two questions here. If fetal placental unit is asked, only what I am about to mention or what I have taught before should be added. And don't confuse that with functions of placenta. Feto-placental unit is only one function of placenta. If the question is functions of placenta, then you need to write about the nutritive role, about the excretory role, about the respiratory role, the formation of various hormones formed in the placenta, and then maybe that feto-placental unit becomes a part of it, right? So when feto-placental unit is asked, please stick on to only this part component. So what happens is during pregnancy, the progesterone and estrogen concentration are increased substantially because that much high level is required for maintaining the pregnancy. Now how are they achieved? During first trimester, the HCG or the human chorionic gonadotropin helps in the maintenance of corpus luteum because that is a major source of progesterone and estrogen. Then later, as the placenta is formed, the trophoblast takes the function of formation. Now see, this picture shows you this is a normal menstrual cycle. During the menstrual cycle, the level of progesterone and estrogen, and this after gestational age shows the amount which is required during pregnancy. So you can clearly see the increase in the amount or the requirement of progesterone or estrogen for maintaining pregnancy, right? So what exactly is feto-placental unit? By 8 weeks of gestation, the placenta has become a major source for these steroids. But remember, placenta cannot synthesize the hormone by itself. It requires help from both the mother and the fetus. So this joint effort in steroid synthesis is what is known as maternal placental fetal unit. The placenta by itself is an imperfect endocrine organ. So it requires help both from the mother and the fetus, and these three in helping in the synthesis of steroid is what is known as feto-placental unit, right? Placenta cannot manufacture adequate cholesterol on its own. So cholesterol comes from the mother and gets converted to progesterone in the placenta. So this progesterone is then returned to the mother 90%. 10% enters into the fetal circulation where it gets converted into cortisol, that is up to 10 weeks, right? So this is very important because not only it will help you in reproduction but also when you are learning about the adrenal gland. So learn the basic. You have cholesterol. From cholesterol you have pregnenolone and progesterone being formed. Then you have the hydroxy component, hydroxypregnenolone and hydroxyprogesterone, and in case of cortisol it gets converted into deoxycortisol and cortisol. As far as we are concerned, we will stick to this. So we have 17 hydroxypregnenolone and 17 hydroxyprogesterone, which is going to form DHEA and androstenedione. From androstenedione you can get testosterone. The presence of aromatase helps in the formation of estrone and estradiol. So kindly learn this chart by heart. Right? So this you can see the difference between the mother, placenta and the fetus and the various hormones being formed there. So just remember, we said the first thing which is required for the formation of estrogen or progesterone is cholesterol. So you have the cholesterol going from the mother circulation into the placenta. The placenta has got enzymes sufficient to form pregnenolone and progesterone. And once this progesterone is formed, it shifts into the mother. And some amount of pregnenolone shifts to the fetus. The growing fetus adrenal gland is able to convert this pregnenolone in its sulfate form to what is known as dehydroepiandrostenedione. If you remember, any hormone during the growth of the fetus is very crucial, amount is very crucial for the development of its external and internal genitalia. So in this, DHEA is converted into a sulfate form, which is an inactive form. Now this DHEA sulfate, again when it moves through the fetal liver, it forms hydroxy component of DHEA sulfate. Now these two, as they cross the placenta, they are acted upon by the enzyme sulfatase and they form DHEA and hydroxy DHEA. And the placenta has got the machinery to convert this DHEA and hydroxy DHEA into estradiol, estrone and estriol, which is finally released to the mother, right? So this is a simple version of the same diagram. So just remember this, how cholesterol substrate is provided to the placenta, the machinery or the enzymes which is present in the placenta which helps in the formation of pregnenolone and progesterone, and how progesterone which is formed in the early steps gets shifted to the mother, pregnenolone gets shifted to the fetus, and there due to the presence of enzymes it forms DHEA and hydroxy DHEA. It cannot be there in the active form because you have the formation of external and internal genitalia happening there in the growing fetus. So it gets converted into its sulfate form. And this sulfate form, due to the presence of sulfatase enzyme, is removed and DHEA and hydroxy DHEA forms estrone, estradiol and estriol, which is then released to the mother. So to conclude, the function of this fetoplacental unit is production of estrogen and progesterone, which are essential for the maintenance of pregnancy, and the second important function is initiation of parturition. So I repeat, when we ask you fetoplacental unit, kindly stick on to the steroid genesis, and function of placenta will include the other functions, be it nutritive function, excretory function, respiratory, formation of hormones, and including the fetoplacental unit. Another important question is the physiological basis for pregnancy test, right? So in this, human chorionic gonadotropin or HCG is the most important hormone here, which is secreted by the placenta during pregnancy. It peaks between 10 to 12 weeks of pregnancy. Now detection of this hormone either in urine or in blood confirms pregnancy, right? So this HCG, initially it is important, as we saw previously, to maintain the corpus luteum so that it forms estrogen and progesterone till the placenta takes over this function by 6 weeks. Now HCG has got two components, alpha and beta. Beta is more specific and used for the diagnosis of pregnancy, right? So the basis for this test, either it can be a biological test or an immunological test. Now if it is a bioassay, as the name suggests, it has to be done on an experimental animal. So this can be performed only after 2 to 3 weeks of consumption, because you require a high quantity of HCG in the urine then, and results cannot be obtained quickly. So these are, you can say, the demerits of using the bioassay system. So what is the basic behind this biological test? You collect the morning urine sample around 60 to 70th day of amenorrhea, because you require high increased quantity of HCG, and 3 to 5 ml is then injected into the peritoneal cavity of the animal. Now the animal can be a rat, rabbit, mice, or even the dorsal lymph sac of a frog, right? So if there was HCG in the urine, it brings about the ovulatory change in the female animal, and in the case of a male animal, it causes the release of spermatozoa. So pregnancy were confirmed initially like this, right? But then we already saw the ill effects of the biological test. Now these are the various biological tests performed on different animals, be it a mice, a rat, rabbit or a frog. At least you need to know 2-3 examples from this and write the demerits of the system. So we move on to the immunological test. What are the merits? They are more accurate, result is obtained quickly, and it can be carried out as early as 5th day of conception. In other words, only a minute quantity of HCG in the urine is required, urine or blood is required. Now it goes by various name, but then if you understand the name properly, it is very easy to follow. So you have first classification, what is known as agglutination inhibition test, right? Now something is going to be agglutinated, but then you say positive or negative according to this agglutination inhibition. Now if you use RBC coated HCG, then we use the term heme agglutination inhibition test. But if we are using latex particle, we use the term latex agglutination inhibition test. Now here this picture depicts it. You have an urine sample and you are adding HCG antibodies to it. Along with that, you are going to add, in this case it is latex agglutination inhibition test. So you have latex particle coated with HCG. Now if the lady was pregnant and she had HCG in the urine, this HCG will act with the antibody, right? Now when I am adding this latex particle, there is no antibody to be added to this latex particle because the antibodies are already bounded with the HCG present in the urine. So the test is a positive when there is no agglutination occurring. At the same time, the test we say is negative or she is not pregnant if agglutination has occurred. So kindly understand this picture, it's very easy. So this comes under agglutination inhibition test. Apart from that, like any other measuring any other hormone assay, if you remember, we have two important procedures. One is the radioimmune assay and the other one being ELISA. So you are already familiar with these two, so I am not going in depth. And then you have the Grab index test, which is the most common and routinely done till now. Here we stopped in the process of agglutination. You have an urine drop, you have HCG present there, and we add latex which contains a HCG antibody. Now due to the presence of HCG antigen, this combines with antibody and we say we get agglutination, so it is a positive test. Remember, this is different from agglutination inhibition test which was mentioned earlier, right? So this is the basis for the Grab index test, which is very simple. So nowadays we have what is known as strip test or the card test. This is a two-side sandwich assay because we use two types of anti-serum HCG. One is in its conjugate form, the other one in its native form. So we say there is a control, there is a pink or a red line, and then if the test sample also shows the same line, pink line, then we say that it is positive or the person is pregnant. Now what is the basic behind this? There is a strip containing a dry conjugate of anti-HCG, right? Now that is dipped in the urine. Now that urine has got HCG, it will react with anti-HCG conjugate and it will form what is known as anti-HCG conjugate HCG. Now this complex starts migrating onwards and there you have another concentration of what is known as native form of anti-HCG. That is why it is known as a sandwich test. You have HCG which is being sandwiched between a conjugate anti-HCG and a native anti-HCG, right? Now this immobilized anti-HCG is able to capture the HCG molecule of migration, causing agglutination. So you get a variable purple band or a pink band along the exact location of the immobilized anti-HCG, right? So no colored band indicates negative result. So the strip which contains both conjugate and native anti-HCG is dipped in urine. So urine has HCG. Now this HCG combines with the conjugate anti-serum HCG antibody. They form a complex. They start migrating above and there you have a native form of immobilized anti-HCG. Now these three together are able to form what is known as a variable purple band, right? In case the urine did not have HCG, then there is no test for the purple color. So this much you need to write as far as this immunological test for pregnancy. It depends upon the mark. If it is for 5 marks, then you mention about the 2 different types and in brief mention about the biological assay, 1 or 2 names, and then go in depth for the immunological assay. Now contraception is another one important question, but here I will just outline the topics under which you need to write. First write the definition, then going to explain that there are two important types, temporary method and the permanent method. The temporary method, you have barrier method. In the barrier method, you need to give examples for mechanical barrier and chemical barrier, right? So where mechanical barrier, you will have the diaphragm and now this thing, while in case of chemical barrier, any anti-spermicidal cream, right? Then you go into explain the natural contraception. So you have three classification under this. It can be either using of safe period, coitus interruptus or complete abstinence, right? Then you start mentioning about the intrauterine device. Under intrauterine device, it can be a non-medicated one like a lipis loop or a copper T, or it can be a medicated one like melena. So you need to write the things under that. Then you go on to explain the hormonal mechanism. Under hormonal mechanism comes the role of pills. So it can be a combined pill where there is contribution of both estrogen and progesterone taken throughout the cycle, or it can be a sequential pill. As the name suggests, it follows the initial few days of estrogen followed by progesterone, or the emergency pill or the postcoital pill. And the latest you need to mention is the slow releasing subcutaneous implant. Example will be your Norplant. Then after that, you need to mention if post conception, what are the measures which can be taken. So it can be either endometrial aspiration or MTP, medical termination of pregnancy. The various methods used, it can be a vacuum aspiration or it can be DNC, which is dilatation and curettage, right? After that, you go on to explain about the permanent method. It can be vasectomy in male and tubectomy in case of females, right? So this covers contraception. Kindly prepare accordingly. So if you can mention few examples along with each different type and the basic mechanism by which they act. Like if you are mentioning about the IUDs, then you need to say how there is a sterile inflammatory process which is happening in the endometrium, which prevents the blastocyte from getting implanted. Or if it is about the action of pills, then you need to mention about how by giving estrogen and progesterone in the form of a pill, you preventing LH surge, so there is no ovulation which is happening, so there is no fertilization and no pregnancy. So write 1 or 2 lines on the mechanism and then follow the same pattern. This I think covers the important question as far as female reproductive system is concerned. The other questions which can be asked, one can be the functions of oxytocin. If it is functions of oxytocin, then you need to write about from where it is formed, and the 2 important function during parturition and function during lactation. So that is one thing which is important. Another thing can be this lactation. So in case of lactation, you need to mention all the 4 different processes, that is starting from the formation of ducts, hormones which are responsible for the formation of ducts, lobules, development of the breast. So estrogen, progesterone along with growth hormone, thyroid, steroids and prolactin, formation of milk, then prolactin has a role, and then you need an expression of milk, then comes the role of oxytocin. So you need to explain that in detail if the short notes is asked on on lactation, right? I think I've covered the important questions of the sequence. I'll be sending a few important questions again with the CR group on WhatsApp. So kindly go through as far as reproductive system is concerned. Thank you.